Hello, my name is Kalpana Katti, and today I'm going to talk about results from our group on the project Design of Humanoid Testbeds of Breast Cancer Bone Metastasis that is supported by the DaCCoTA Ready to Go pilot grant. This is a collaborative work between NDSU researchers and Dr. Anu Gaba at the Roger Maris Cancer Center at the Sanford Hospital in Fargo. So breast cancer is the second leading cause of cancer-related deaths in women in the United States. According to the World Health Organization, there were about 2.26 million breast cancer cases worldwide in 2020 that resulted in 685,000 deaths. So in all cases where deaths occurred in the patients, it was almost always due to the process of metastasis in which the cancer translocates to a distant location in the body and colonizes there. For the case of breast cancer, bone is the most common site for the cancer metastases. So when the breast cancer metastasizes to the bone, it is incurable and the anti-cancer drugs are quite ineffective. In addition, it is also difficult to obtain large number of samples from patients at the bone metastasis condition because at this point, the patients are often in a hospice situation. The traditional cancer models, in vitro models that are reported in literature create some of the characteristics of metastasis, such as hypoxia and some biochemical cues, but they do not recapitulate the entire mechanical and biochemical characteristics of the bone environment at metastasis. Animal models in literature have often failed because the animals die before metastasis to bone occurs. So thus, there is a great deal of need to develop reliable, robust, and humanoid 3D in vitro models of breast cancer bone metastasis. To that effect, we have developed a novel nanoclay polymeric composite scaffold system and using tissue engineering approaches have been able to build bone tissue on this scaffold by seeding the scaffolds with mesenchymal stem cells. Once the extracellular matrix is observed on these bone scaffolds, we seed them with breast cancer cells, at this point, commercial cell lines of breast cancer. And we were able to enable a development of mesenchymal to epithelial transition stage bone metastasis of breast cancer. So in this project, what we're trying to do is to use the breast cancer tissues from patients, generate cell lines, and seed them onto the scaffolds in order to create bone metastasis cancer tumors on the scaffolds in an in vitro system that are truly humanoid and they are patient-derived. So we have been able to obtain 7 patient tissues of breast cancer from the Sanford Hospital under the supervision of Dr. Gaba at the Sanford Hospital. We have developed a protocol to generate tissue cell lines from these samples that are obtained. Once these cell lines are generated, we seed them onto the scaffolds. And here are some photos here of showing scaffolds that have been now seeded with patient-derived cell lines. Using this process, the protocol developed actually involves sort of preconditioning the cancer cells with mesenchymal stem cells prior to seeding onto the scaffolds. And using this protocol, we have been successfully able to generate bone metastasis breast cancer tumors from patient-derived tissues on our bone scaffold systems. In the future, we plan to investigate further the use of this for personalized medicine and also for the discovery of biomarkers of bone metastasis of breast cancer. Thank you.